Thank you very much for that introduction. Can everyone see my screen? I just have kind of the title slide here. You're good? Okay. So thank you. This is one of my favorite conferences every year. I'm very, very sad that we're not all up in Montreal enjoying beautiful culture up there, but I'm glad we can all come together over Zoom. So kind of the abbreviated title of my talk today, I'm just calling Beyond Reference Genomes. Historically, my lab has done a lot of work on genome assembly, going back nearly 20 years with Sanger sequencing, 454, Illumina, PacBio, Oxford, basically every major platform I've been very excited about to establish reference genomes. And we've done this in many different species, plants and animals and fungi, basically across the tree of life. Today, though, I thought what I would do is talk about, some of the work that was going on in our lab kind of beyond assembling individual genomes, talk about some of the algorithms, the data structures, and the systems that can sort of take some of these technologies and apply them in a more translated way. So specifically, these days, I spend a lot of my time kind of broadly in translational work, where I'm trying to use sequencing and computational methods and quantitative methods to study, say, human diseases. So here are kind of two example pedigrees that we've been studying, looking at developmental disabilities, different forms of cancers. And then as you heard, we also spend a lot of time in different agricultural systems where at some level we're trying to answer the same questions, although obviously the systems are very different. And sort of the broad overreaching goal for all of this work is sometimes called genotype to phenotype, where we can do all the sequencing, we want to do detailed comparisons, and really what our goal is to identify those specific variants that are going to give rise to these different diseases or traits of interest, to really be able to pinpoint them. So this is more than sort of establishing a reference genome, sequencing a single genome. This often involves, sequencing families, large pedigrees, large populations, and doing very detailed comparisons across them to try to identify these types of variants. One of the major things that we bring to the table is, of course, lots of other groups are working on this problem and lots of different diseases. And I think our expertise really comes into play for these so-called unresolved, unsolved different characteristics, different traits. So these occur for potentially a variety of different reasons, but kind of the scenario is, there'll be a first look at a system, maybe nothing really emerges that really explains the disorder, and you need to kind of consider, why is it that we can't resolve these different traits? For a lot of the projects I'm involved in, our central hypothesis is that these different diseases, these traits remain unsolved because they involve variants that are just inaccessible, using standard approaches that have been previously applied. So our goal is to kind of bring some new technologies, both sequencing and computational, to try to make these inaccessible variants visible and then hopefully interpretable so that we can identify the sources of these different phenotypes. So what makes a variant inaccessible? There's many different reasons. Let me just sort of give you a few of the ones that come to mind. So number one on my list would be these are just complicated variants. So standard approaches, and by that I really mean short read sequencing, are superb for identifying single nucleotide variants over most of the genome. There are regions that, because of repeats, remain accessible. There are some systematic biases with Illumina sequencing, notably in GC rich or poor sequences. But by and large, single nucleotide variants do well, but more complicated variants remain very challenging to identify. And it gets even more so when there's a lot of variability, where from one sample to the next, there's a lot of mutations. They're just complicated variants. It's just very difficult to identify them, pin them down using standard approaches. So what we really need to bring to the table are new technologies for more comprehensive and more accurate variant identification. Kind of number two is maybe we can find the variants, but they have very sort of complicated inheritance. Maybe these are de novo mutations. Maybe these are somatic mutations. Maybe these are mosaic variants. Maybe these are private variants that are only observed basically in one family on the whole planet. So we just really need very careful ways where we can compare variants amongst different samples, across families, across pedigrees, so we can do very, very accurate identification of these variants, and then hopefully sort of pinpoint, variants that are specific to those individuals that carry the phenotype and not present or present only very, very infrequently in other individuals. And then finally, what makes it inaccessible is there's just a very complicated mechanism. this could be non-coding variants. These could be sort of distant enhancers, different distant trans-eQTLs. They could have very small effect sizes. They could have epistatic or epigenetic interactions. And I think the way we're going to make progress on these is through the analysis of very large populations, the integration of multiple data types, where we can kind of tease out these really subtle interactions in a very sophisticated way. Now, I'll be the first to admit these are hard problems. The older I get, the more I realize that genetics is hard in the sense that in a given system, there could be many different proteins, cellular aspects, developmental aspects that all come together. So it's very hard as an engineer. What I want to do is sort of isolate individual components and study them. But these systems are very complicated in the sense that it's sometimes very difficult to isolate them. I predicted 1,000 years. We're still going to be struggling with a lot of these issues, especially for variants for systems that are more complicated, that have really subtle interactions. These are just really, really difficult phenomenon to study. But what I wanted to do today was sort of describe some of the latest advances, some of the latest work we have in bringing new algorithms, new data structures, new computational systems to try to address these problems. Specifically, what I'm going to do today is describe 3 new algorithms, 3 new systems that we've developed that try to sort of focus on different aspects of these complex variants, these complex unresolved variant types, and hopefully make some progress here. So what I'm going to do is just sort of walk through them. They're related in the sense that they all kind of work together. They all play with each other over time to bring forth new information, new knowledge to hopefully resolve some of these very complicated variants for these very important diseases and traits. So the first of which is a new algorithm called Jasmine. What it's really designed for is fast and accurate structural variation comparison across many individuals. Before diving into it, I really want to acknowledge the lab members that have really led these efforts. Number one here is Melanie Kirsch. She's an amazing programmer, amazing algorithmicist that has sort of developed a lot of these implementations, come up with a lot of the ideas behind them, the structures. There's some very sophisticated work, as you'll see in a second. In addition, she's joined by my postdoc, Sergey Aganov. He's a great mathematician by training, moved into computational biology, and just brings a lot of resources and skills to the table. And then also a lot of support from Rachel Sherman, who's a PhD student in Steven Salzberg's group that has also been really, really helpful in pushing forward a lot of these algorithms, a lot of these ideas. The source code for this work, and actually all the work I'm going to talk about, is available on GitHub right now. You can go check it out. So a lot of the work that we're focusing on in my group is looking at structural variants in a lot of different systems. I wasn't going to really talk about it today, but we had a major paper come out about 2 weeks ago where we're looking at structural variants in tomato genomes, but kind of the conclusions hold true in basically every system that we look at, which is that structural variants, so these are variants that are 50 base pairs or larger, this could be insertions, deletions, inversions, translocations, and so forth, are proving to be critically important for a lot of different diseases and phenotypes. In this paper, we used long-read sequencing, Oxford Nanopore long-read sequencing, to sequence 100 different varieties of tomato genomes, from which we uncovered more than 200,000 structural variations across the population. In terms of their sort of origins, a lot of them came from what are called transposable elements. These are sequences that can, so-called jumping genes that can move around inside of the genome. We found kind of a really interesting patterns there. Sort of genome-wide, we were able to correlate and associate these structural variations with changes in gene expression in many different contexts. Some of these were pretty straightforward. If there's a big deletion over a gene, yes, its expression tended to go down. If there's a duplication, yes, the expression tended to go up. But there's a lot of sort of subtle relationships that we detected as well, where deletions, insertions, inversions, other sort of complicated variants within, say, introns or in flanking promoter regions did tend to lead to changes in expression. And then furthermore, beyond just these associations, we were able to connect some of these structural variants with a few different major traits. So the productivity of the plant, the size of the fruits, the flavor of the fruits. I really am convinced that this is going to set sort of a new paradigm for trait associations in plants, in humans, in animals, across the tree of life. I think it's really now time to look at structural variants. Broadly speaking, there are 2 major ways to look for structural variants. And in fact, in the tomato paper I just described, we use both approaches. One is alignment-based strategy where we're going to take reads. What I really like for this these days is long-read sequencing, We're going to take those long reads, we're going to map them from a sample, we're going to map them to the reference genome, and sort of conceptually, we're going to look for split read alignments. You can imagine half the read aligning here, half the read aligning over here. That can be, depending on exactly the framework, that can be indicative of a deletion, that can be indicative of, say, a translocation across chromosomes. It could be indicative of an insertion, inversion, other sorts of structural variants there. There's a lot of advantages to mapping. It's very fast. We can accurately identify most variants. It's very powerful for heterozygous variants. Kind of the main limitation there is that very long insertions are going to be lost, in the sense that if your insertion is longer than your read length, you can notice half my read aligns and the other half kind of doesn't, but you're probably not going to be able to span the whole insertion there. We noted that in Tomato. We noted that in other projects. It's something that we continue to work on by integrating de novo assembly with mapping-based approach. The flip side to this is de novo assembly, where we just take our reads from a sample, we assemble it up into hopefully very large contigs, map those to a reference genome, and then whenever you see a variation there, we get some confidence that there's a structural variant. There's some practical challenges for this. De novo assembly is considerably more computationally demanding. You also need often higher coverage. You need longer reads to make this successful. You're going to be limited by your contig length. The advantage, though, is in principle, this can capture all types of variants. Another sort of challenge, though, is heterozygous variants are very challenging. And also in the here and now, I would argue that the way that we represent a de novo assembly is somewhat limited, in the sense that you get a consensus sequence, but you often don't really know what was the supporting data that led to that sequence. So if there are consensus errors, those are hard to identify. If there are structural errors in your assembly, those can be hard to identify. What you really want is sort of, what is the supporting evidence for this variant that has been identified, which to me, sounds like you want to be able to look through a multiple sequence alignment like you would get from an alignment-based approach. So there are pros and cons to both. In practice, you're probably going to want to do both approaches if you want to have the most complete representation. Although in the here and now, I would say alignment-based approaches are a lot more practical, a lot more efficient, and tend to be more sensitive and accurate. For structural variation detection using long-read sequencing, the methods that we really like that came out of my lab are a pair of algorithms, NGMLR, and for long-read alignment, and a structural variant caller called Sniffles. These are published a couple of years ago. The lead author was Fritz Setzlack. He's now an assistant professor at Baylor College of Medicine, continues to do amazing work there. I'm guessing that these are pretty well known to this audience, so I wasn't going to go into them in a lot of detail into how they work, although please go check out the paper if you want to learn more about them. Since the publication of the paper, we've been continuously trying to improve on these methods, continuously trying to make them faster, more accurate, more sensitive, and a really good resource for this has been the Genome in a Bottle project. I'm sure this is very familiar to everyone on this call. So specifically what we've been doing is through the Genome in a Bottle, there's a number of samples that have been sequenced that are so-called trio, where it's a mother and father and an offspring, all from the same family. And what this allows us to do is do a genome-wide assessment of the accuracy of the approach. And kind of the gist of it is something called Mendelian concordance or Mendelian discordance. And kind of the conceptual framework is if we can identify variants in the parents, we should be able to observe many of those or in a very predictable way in the offspring. And then if we observe any variants in the offspring that are not present in the parents, the vast majority of these are going to be false positives. It is true that from generation to generation, there are so-called de novo variants that just sort of spontaneously occur. This has been pretty carefully measured for single nucleotide variants, and best reports estimate maybe 100 single nucleotide variants genome-wide from one generation to the next. In terms of structural variants, I think there's a lot less literature around this. There's a lot less reports of it. They do occur, for sure, but I would say they should occur, less than single nucleotide variants. Certainly less than 100, on the order of tens, maybe even single-digit de novo variants in healthy samples. Now, of course, if there's a genetic disorder, especially if there's cancer or some other genetic abnormality, there could be many more. But I think these should be quite rare in healthy individuals. So because of this, this lets us get at, in a genome-wide, in a systematic way, lets us do analysis as to what is the false positive rate of variants called in this way. So if you just do this using different data sets, so this is looking at the hi-fi data from Genome in a Bottle using best approach, we get a pretty good discordance rate. Out of the tens of thousands of variants are called, only say 7% of them are discordant. So these are only observed in this example in the son, not in the parents, which is pretty good, but is very challenging when you're doing a disease study where you basically only expect there to be one variant that's relevant, but here you're getting thousands and thousands of false positives. That's going to be really problematic to any sort of downstream analysis that you've been doing. So we've been trying to systematically dissect what are the sources of error, how can we combat them, how can we improve this to get it down from, say, thousands to maybe a few hundred, of which many of these could actually be real. So we've identified three major issues that lead to sort of false positive calls in these studies. Let me kind of give you kind of a quick tour through what we've noted about them. So the first is seemingly very mundane, but is actually the largest sources of problems. So anytime you run a variant caller, if you run freebase, if you run deep variant, anytime you run any sorts of variant caller, if you run Sniffles, there's a series of sort of thresholds that are used to evaluate, is the variant there or is it not? For structural variants, you get, in addition to kind of the regular ones about how much coverage do you have, you also get different thresholds over the variant representation itself, especially if you're using long-read sequencing that does tend to have a higher error rate. Maybe in one sample, an insertion is reported as 29 bases, and a different sample is reported as 30 bases. If your cutoff that you're using for sensitivity is 30 bases, in one sample it'll be found, in the other sample it will be not. So as you're, in a single sample, that's perhaps the best you can do, but if you're trying to do any sorts of comparisons, this can lead to these so-called thresholding effects, where a variant is found in one, not found in the other. We need to be able to address that. So fortunately, there's a pretty straightforward solution for this, is to call variants in a very, very sensitive mode up front, do your comparison across sensitive variants, and then at the end, you can sort of post-hoc filter to variants of a certain size or a certain level of coverage to try to combat that. So that's kind of the most straightforward, although is the largest source of Mendelian discordance when we look at it. Another error is when we're using long-read sequencing, there's quite a bit of noise in the sequence, which can lead to little shifts in the position of the breakpoint, or maybe the sequence fidelity there. And our approach for combating that is to apply very localized, like little teeny tiny de novo assemblies around these variants in order to sort of, best as we can, resolve the breakpoints, resolve the sequence that's there. For this, we have a new pipeline called Iris that's very efficient. It sort of leverages sort of standard approaches from de novo assembly, but as a way, in a very automated way, you can take sort of a first pass, sort of rough cut set of variant calls and give you very pristine variant calls with the sequence resolved, the breakpoints resolved with very high fidelity. And then finally, the big challenge that we've been really putting a lot of efforts into recently is just there's very inconsistent merging methods. There's a package called Survivor that a lot of people use. In fact, also comes from Fritz, and it's very effective overall. But as you really zoom in at some of this discordance, you notice these methods for identifying variants do have a number of limitations. There's other packages that attempt to do this. I would say the most common strategy is conceptually, we're going to take our set of variants, we're going to sort them by chromosome position and do kind of a little on-the-fly clustering along by chromosome position. And that works pretty well, but there is this remaining set of variants that remain confusing if you try to do this. And a lot of it, again, comes from a little bit of noise in the sequence that resolves. There can be technical noise, but there can also be biological noise, whereas you're comparing, say, variants in one family to variants in a different family. It may not have exactly the same breakpoints, may not have exactly the same sequence, but in terms of the phenotypes that can arise, at some point, they're going to be so similar, you want to be able to reconcile that. So we need really fine control over the types of variants that are going to be merged. We need this to be done in a very accurate and robust way. So our approach for this that Melanie has really spearheaded is called Jasmine. The input to this is going to be multiple VCF files. It's predominantly for structural variants, although you can also use it for indels. You could do it for SNPs, although it'll be it'll sort of not take advantage of many of the features that are present there. And then it's going to be able to take these variants and then combine them in a very powerful way. So the strategy for this is, rather than just sort of sorting them by chromosome position, we're going to take into account, because it's so focused on structural variants, it's going to take into account both the position and the length of the variants that are present there. And it's going to sort of project this into this 2D space. And then inside of this 2D space, we're going to do on-the-fly clustering to be able to find those sets of variants that are all sort of systematically compatible to each other. This is going to be using something like a maximal spanning tree algorithm in order to find these components that are compatible with each other. Once we have it, we can output a merge VCF file that is sort of very going to carefully and systematically resolve the types of variants that are present. And just one slide, there's a number of specialized requirements that we're going to use here. You know, a lot of tools, including Survivor and others, have a tendency to, if you have multiple variants in one sample, we'll just sort of merge them together on the fly. Sometimes this is useful, but we find sometimes this can confuse a lot of the downstream analysis. So we're going to do sort of careful accounting of where the different variants come from. And then to keep the memory requirements small, make it very efficient, we're going to be using an advanced data structure called a KD tree in order to represent these variants in two-dimensional space so we can query it very, very efficiently to be able to find variants that are compatible with each other. There's a bunch of other improvements as well to make this go fast and efficient. So conceptually kind of putting all these ideas together, we can align with NGMLR, we can call using Sniffles variants in this sort of sensitive and specific mode, we can refine the breakpoints, and then we can clean up in the merging using Jasmine, using those advanced data structures and techniques I just described. So what does this do? Instead of having 6 or 7% of false positives, now we have our Mendelian discordant rate down to 0.36. This is just a few hundred variants per sample. This is kind of in line with the number of de novo variants that have been reported using single nucleotide variants. We're kind of in the right order of magnitude. As we investigate the remaining discordant variants, what we're coming to realize is the initial variant call set needs to be very carefully tuned to the type of data that you have available. Without going into a lot of detail on Sniffles, there's a number of parameters that need to be very, very carefully tuned depending on the type of data. Sniffles was really originally developed primarily around PacBio long-read sequencing, but as you go into HiFi data, as you go into Oxford data, there's a series of parameters that need to be adjusted because of the different error characteristics that are present there. So we've had these new pipelines, we've been applying them to many different samples. We use them in the tomato project that I briefly described. And another project that was actually just accepted at Genome Research about a week ago was looking in breast cancer samples to be able to find structural variants that had gone missing using short-read sequencing. Here is just one of the many examples we found. This is a structural variant identified in the famous BRCA1 gene that is only detectable using long-read sequencing, not detectable using short-read sequencing. So in the same way that we had found variants that were that we could prove in agricultural systems as being related to different diseases and systems, I'm very excited to use long-read sequencing and these algorithms for human diseases as well. You can read more about that on our bioRxiv preprint as well. So just to sum up for Jasmine, this enables very accurate structural variation identification and comparison. We're developing sort of specialized pipelines depending on the types of data that you have. As much as I'd love to give you just one push button solution, the reality is they have different error models, they have different characteristics that need to be tuned. It'll mostly just be sort of parameter tuning to make this as robust as possible, but this is producing a panel of really high quality structural variants that we can use for screening different disease systems, different populations very, very rapidly. Okay, so that's sort of Jasmine, where we can very accurately identify structural variations out of an existing data set. The next sort of system I want to talk about is called UNCALLED, which is if you have sort of regions or genes that you already want to go look at, and you want to be able to scale this to many more samples, UNCALLED is going to make this possible to do it very, very rapidly and very inexpensively by a computational enrichment of targeted nanopore sequencing. This is really the brainchild of my student, Sam Kavaka. We had great support from Jan Finn from the Winston Timp lab, and then also Bahan Ni from my lab and Alexis Battle's lab to be able to make the software. Software is available on GitHub to go check out and run right now. So the sort of, as I was kind of alluding to, the scenario for UNCALLED is often when you're doing shotgun sequencing, a lot of the sequencing reads are kind of wasted in the sense that they come from regions of the genome that you're not really interested in. So, using Jasmine, maybe you'll sort of narrow it down to these are the 100 variants I'm really interested in, but now I need to go screen them in thousands and thousands of other samples. Yes, of course, you could do whole genome sequencing to look over thousands and thousands of samples. That becomes very expensive, becomes very time consuming, and you're just gonna be collecting a lot of data that you're just not really interested in. Another important scenario, especially now in the age of COVID, is maybe there's a pathogen that you're really interested in, like the COVID genome, but to get at it in a meta-transcriptome, you're gonna be sequencing, human RNA, you're gonna be sequencing, the vast majority of which will be human RNA, only a subset of the molecules are gonna be from the viral genome. Wouldn't it be great if we could enrich for those viral genomes on the fly without having to worry about the human RNA that is going to be present there? So those are some of the sorts of scenarios that this sort of approach can really be powerful for. There are experimental methods, of course, that help do enrichment. By far the most popular is PCR, where you can kind of amplify regions of interest. This is very robust, it's very reliable, but it has many limitations. You can typically only target really small regions. Every region that you're interested in requires a separate reaction with different primers, so it just requires a lot of design, a lot of reagents, a lot of hands-on time if you're going to try to scale this up. For nanopore sequencing, there is also an experimental protocol that uses CRISPR-Cas9, where you cut out the regions that you're interested in, and then you can kind of do selective sequencing over just those regions. This works great if you have, say, 10 genes that you're interested to look at. But as you get into larger and larger sets of genes, or you get larger and larger regions of the genome, this becomes just a lot more reactions that all have to work. Every time you have a new panel, you have to design new guide RNAs. So there are some inefficiencies if you want to try to use this. A new approach that is unique to nanopore sequencing is to do on-the-fly enrichment using something called read-until sequencing. So the way nanopore sequencing works is there's a molecule of DNA or RNA that is passing through a pore. As the molecule is passing through, you get signal data associated with what's the nucleotide sequence. Remarkably, this is done in real time, where at about 450 base pairs per second, you get that signal data. If you can somehow understand that signal data and decide if is this signal that I'm interested in, is this a molecule that I'm interested in or not, there's an API call that you can reverse the voltage of the pore and selectively change it, spit out the molecules that you're not really interested in. This is really, really specialized for nanopore sequencing. Would be impossible with, say, Illumina or even PacBio to do anything like this. There have been previous reports using read-until sequencing, but they've been pretty limited. They didn't really produce meaningful enrichments. There is now a new preprint from Matt Luce's lab that is able to do this at pretty nice scales, although it requires GPUs, has some other limitations I'll talk about later. But I think that we're starting to see a resurgence of these methods thanks to new algorithms and also thanks to improved efficiencies with the nanopore device itself. So the system that we've developed is called UNCALLED. It has kind of a cute backronym, utility for nanopore current alignment to large expanses of DNA. But kind of the headline is we're going to take this raw signal data. We're going to do something like very, very lightweight base calling, in the sense that we're going to go from signal data into nucleotide sequences by sort of a statistical analysis of the signal data. We can then take that nucleotide sequence and then use an FM index to do on-the-fly alignments. Although to make this really efficient, we killed ourselves to make it streaming, to make it very efficient. There's a lot of sort of nice data structures in order to represent sets of alignments that are potentially discontiguous very, very efficiently, both in time and space, to make this practical. We have a preprint describing it that I'll talk about some of the main results here. So there's two major modes for UNCALLED. You can do so-called enrichment. So if in your sample there's genes or other sequences you want to go sequence, we're going to do mapping. If it maps to those regions of interest, we'll keep it. If it does not, we'll eject the molecules. But there's also depletion, where if we're sort of in our sample, if there's certain regions or certain genomes that we're not interested in, if it maps to those regions or genomes, we'll split the molecules out. Otherwise, we can accept it. They both rely on the fly mapping. They both rely on ejection, but they kind of, are you ejecting because it's something you see that is known, or rejecting because it is unknown, essentially. So as a baseline, we were applying this to a metagenomics sample. It's a mock metagenomics sample called, from a company called Zymo. It has a number of bacterial and fungal species. First experiment was not read-until sequencing, but we took an existing signal data from an existing sequence data set that had been base called and mapped with Minimap2, and then we took those same signal data, apply Uncalled to it, and kind of the punchline is we get basically exactly the same results. For every read that has been mapped either in signal space or using Minimap2, basically every read can be accounted for which genome it comes from. The next experiment, which I think was a lot more exciting, was this is what is plotted here is sort of the natural distribution of molecules in the sample. What we want to do here is take those bacterial genomes, decrease their abundance, and that yeast genome increases abundance in a totally computational way using Uncalled. So we did this. I'll just kind of give you some of the headlines. The accuracy was excellent. We had over 96% accurate ejection rate compared to Minimap2. And then we had over 99% acceptance rate that we kept those molecules from those yeast genomes that we wanted to do. So in terms of output, we increased the amount of sequence data from the yeast genomes by 4.5-fold. We dramatically decreased the amount of off-target sequencing. We just sort of dramatically expanded the amount of the sample that was the yeast genome. So I think this is just kind of an amazing result where, thanks to software, purely software methods, we can kind of sort of skew the distribution of the molecules and skew the distribution of the data that are actually coming out of the instrument. There's just endless opportunities for this. The next experiment for this was to apply it in breast cancer. So there's a company, Invitae, that has a panel of 148 genes that they have identified as being extremely important for hereditary breast cancers and other and cancers of other systems. And what we're going to do here is normally if you do whole genome shotgun sequencing on a MinION, you only get a few X coverage sequence data. What we want to do is really concentrate all that data across these human genes so that we could do detailed analysis of the variants that are present there. So we did this. One thing that you really have to pay attention to is with MinION sequencing and also Promethean sequencing, your active pores does decrease over time. They just get clogged, they get stuck, they get sort of fried out. But there's a protocol where you can flush the pores using a nuclease that sort of chews up any sort of DNA that's in there, reload them. This restores the activity of the pores so that you can then get good yields coming out on the back end. So doing this for real, in an experimental way, we did it sort of side-by-side from the same sample of DNA. Over the sort of control run, we got about 5x coverage across the whole genome, across the genes of interest specifically. But thanks to Uncalled, we could skew this distribution, shift it up, and get about 30x, just shy of 30x coverage over the genes of interest from this cancer panel. When we look at what sort of results can you get using 30x coverage from Uncalled, we get very, very high concordance for, say, SNP and indel calling. We get 98 or so, 99% accuracy for SNP calling, a bit less for indel calling just because the data are noisy. Critically, though, for structural variant calling, we get dramatically improved sensitivity. We had 100% concordance of the variants that we could call out of our enriched data compared to more expensive, more difficult whole genome data using either HiFi or Oxford Nanopore. Critically, we detected about twice as many structural variants using the Nanopore data compared to whole genome Illumina sequencing. And then finally, because it's Nanopore data, you can also go do a second pass look at the signal data to be able to detect methylation. We get extremely high concordance, 96% concordance to say either bisulfite sequence treated or whole genome shotgun Nanopore sequencing data. So this is a great opportunity where we can take a certain amount of capacity on a MinION device and then basically skew it to whatever sort of regions of interest, get really, really robust, really, really accurate results on the back end from that. We have a preprint that describes these results. Since then, we've been working hard to scale it up. In the preprint that's out there, we said about 100 megabases was the effective practical size, although we've smashed through this limit thanks to Sam and Bohan's great work. we have now been able to do through that great. We've been able to enrich much larger gene panels. We've been able to get great enrichment over entire human exome using this. So we're really excited about this moving forward. We would love to use this for RNA, especially for, say, COVID sequencing. That's what we're working on now. There is a different sort of Kamer model about how nucleotides passing through the pore give you different signal characteristics. That's what we're trying to learn right now. That requires some optimization. And there's other further optimizations that we plan to do in the near future. So now we can, from Jasmine, we can accurately call variants. From UNCALLED, we can sort of accurately move this to larger populations. And then the last thing I really want to mention is, how are we actually going to do analysis over huge populations, like hundreds of thousands of genomes? And that's where new advanced computational systems, advanced algorithms, and advanced data structures are really going to make an enormous impact to the world. So to focus in, I'm going to talk primarily about a system called Sapling. This was developed also primarily by my awesome student, Melanie. And we also got great support from another student, Aran Das, in the lab. To put a little context on this, I'm very involved in a big NHGRI project called AnVIL. And this is a cloud-based system for doing very, very scalable genomics, primarily for human genetics, although it has capabilities for other systems as well. It's cloud-based, so it basically has infinite scaling. We provide security so that you can do analysis over protected data sets. If you ever worked with dbGaP data, you know what a hassle it is to get all those credentials in place, get all those security privileges in place. Well, we've taken care of that all for you. And then crucially, and most importantly, it's a collaborative environment where lots of people can work together on different systems and tools and share results in a very efficient way. Effectively, what we're trying to do is invert the model for genomic data sharing, where instead of everyone having to download data to their local cluster, do local compute, we've established this AnVIL system that has a lot of familiar tools to be able to do analysis in the cloud so that we can invert the model. We can build the system in the cloud, researchers can go there. Today in AnVIL, this has lots of familiar things like Jupyter, Bioconductor, Galaxy. It also has really great support for batch workflows using something called Terra from the Broad, sophisticated data modeling from Gen 3 from the University of Chicago, and DocStore for dedicated workflows that are going to run at scale from Santa Cruz. All of this lives inside of a FSMA certified environment so that we can work with human data sets, make sure it's protected for great security. This is really what I wanted to highlight. In AnVIL today, there's about 72,000 whole genomes available there over many different consortiums. If you've ever heard of the Centers for Common Disease Genetics or Centers for Mendelian Genetics, this is where all those data are moving forward. Which really kind of begs the question, well, how are we going to do analysis over hundreds of thousands of genomes? If we're going to do this in a cloud environment, the meter is always running. Some of these workflows cost tens of thousands of dollars, hundreds of thousands of dollars to do one analysis. So anything we can do to make this more efficient, more both in space or time, is going to be a huge win for genomics in a very practical sense. And furthermore, it's going to let us do analysis that's never been done before as we can scale into larger and larger and larger data sets. To address this, of course, there's been a lot of great systems that have been developed for alignment, for variant calling, for all kinds of integrated analysis. Today, I'm really going to talk about some of our latest work looking at alignment, and I'm going to describe our system Sapling that is set up to sort of turbocharge a very powerful, commonly used data structure called the Suffix Array. So I'm sure for this audience, the Suffix Array is very familiar. It's basically the sorted list of all the suffixes that are present in a sample and say a reference genome. It's very fast, it's very space efficient, and very very powerful overall. Crucially, because of a suffix array, if you're trying to do a lookup, like the nucleotide sequence C8A in a big reference genome, all of those matches are going to come up in a contiguous set of rows inside of the suffix array. The way that you do this is something like binary search, where you take your query like C8A, you compare it to the middle of your sort of active range in the suffix array, you can decide is cat bigger or less than that, and then sort of iteratively work your way down to find the set of rows that have this match here. Now, because it uses binary search, in principle, this should have really nice scaling properties that in theory, it should scale linear with the log of the size of the reference that you're looking at, although in practice, it does not. And I encourage you to go test this out for yourself. As you get into larger and larger genomes, it does not have linear scaling or does not have logarithmic scaling. It's much worse than that. And that's because in practice, these systems are built on real hardware, real computing hardware, where there's a hierarchy of response times. You know, yes, the CPU can be blazing fast, working at gigahertz speed, but the data that has to go into the CPU has to come from someplace. And as you get into larger and larger data sets, the latency and also the throughput goes down. So that's why it does not have logarithmic scaling to use binary search as you go over larger and larger data sets. So the idea behind sapling, and as you'll see, a lot of different lookup systems is in a suffix array, there's an algorithmic approach to go from potentially a huge set of possibilities to take us to a narrow set. Well, what if we could replace that algorithmic approach with an arithmetic approach that is basically going to give us a prediction about, from this, huge set of possibilities to the set of rows that contain the results that we're looking for. Crucially, we don't necessarily need to find exactly the right answer. Maybe we just kind of need to get to the right region, and then we can do a localized search using a binary search or related processes to kind of refine that. What we're really trying to avoid are those big expensive lookups where we have to go to disk, where we have to go to main memory, that are just going to kill our performance as we go to larger and larger data sets. So this is an instance of something called a learned index structure. This is a brilliant idea that comes from researchers at MIT and Google. They had a nice Sigma paper about this about 2 years ago. And kind of the observation is for many data structures that we use in genomics, many fields, where you have to do an algorithmic approach to find some results, well, maybe we can sort of turbocharge that algorithmic approach using some sort of data model. That's the learned index structure to sort of turbocharge it. So we're going to take a suffix array lookup that is normally an algorithmic task, and we're going to turn it into a prediction task. The plot on the right is a real plot of the human genome, where on the x-axis we have sort of the Kamer code. You can just sort of take all the nucleotide sequences, convert it into an integer. And then on the y-axis, we have its position along the suffix array. This is like the easiest prediction problem there is in machine learning. It's very close to a straight line so that we can get, if we know what the Kamer code is, we know what CAT is, we can very accurately predict where in the suffix array such a sequence might exist, which just makes this very, very powerful. This is what we observe in the human genome. If you check out our preprint, we did this in many other genomes. They all have the same characteristic shape to them, which is very encouraging to be able to apply this very generally. We attempted a couple different strategies for learning that relationship. We did attempt an artificial neural network. And then also because the curves are relatively simple, we also considered a piecewise linear architecture. It turns out the piecewise linear is superior in many different ways. So that's the version that we actually went for our final implementation. Go check out the preprint to go learn more about the specifics. Oh, we also had some optimizations. Again, maybe we don't necessarily need to find the exact row. We just need to get close enough. We had some optimizations to handle sort of these. There are a few sequences that are more difficult to predict. So we kind of we optimize for the common case, and then we can fall back to a slower case for those oddball examples. So this is kind of the main result here. This is in human, where we're matching 5 million queries. Again, we're looking for exact matches using a suffix array, where we can sort of, a parameter to sapling is how much overhead do we want to have. We can have, very minimal overhead, 0.1% space, 10% overhead. But the kind of the nice result there is as we add more space by having kind of a more accurate data model, we can improve the runtime to be able to do these suffix array lookups. We can substantially outperform an optimized binary search. We can outperform Mummer4, which uses a sparse suffix array. We can even outperform Bowtie, which I'm always incredible to say, where just the suffix array for this particular application is more efficient than using a straight ifm index. And then furthermore, this scales really nicely as we move into larger genomes, where we can dramatically accelerate these queries by using this data model, which avoids more and more of those very, very expensive lookups back to main memory or back to disk. So sapling is, I'd say, a really exciting technique because, yes, this is for suffix arrays, but this general paradigm of turbocharging our data structures with a learned index structure, I think is going to be a really important component of genomics in the future when we're looking at hundreds of thousands of genomes when we're doing these massive comparisons. We have a preprint. The source code's available. I encourage you to check it out. But I'm looking forward over the next few years to turbocharging basically everything we do with this source of strategy. So just to sum up, using great algorithms, data structures, new sequencing, we're just finding this universe of all these complicated variants. Hopefully we're going to be able to use them to be able to make great discoveries as to the genetic origins of many traits, many diseases, hopefully lead to a lot of new insights about the world around us. There's lots of people in my lab and our great collaborators that I want to thank. Throughout the talk, I try to highlight them, but this really took great teamwork. I'm always looking for great students and postdocs, so if you're interested in this type of work, please reach out. And finally, I'd like to acknowledge the NSF, NHGRI, the Mark Foundation, and Bloomberg Professor Program for all the funding and support for this work. Thank you. I'm happy to take a couple questions. Sorry I went a little bit long. Thanks, everybody.
Great. Thank you so much, Mike, for that wonderful talk. We've got a couple of questions already, but I'll encourage folks that if you have them to type them into the Q&A. So the first question I have is from one of the panelists, and this is going back to UNCALLED. So great idea to eject molecules from regions of disinterest. What if the molecule is 1 megabases long? Does ejection only work with smaller DNA molecules?
Yeah, let me, I'm just gonna stop sharing. Yeah, so great question. So, so the molecule is passing through the pore, and it'll immediately in real time, we're gonna get signal data. In sort of a standard configuration, you get signal data every second. So every second you get about 450 observations of the data, about 450 nucleotides worth of data. That's immediately processed by UNCALLED. So even if your molecule is a megabase long, you're going to get the first 450 nucleotides, the next 450 nucleotides, and there's a streaming algorithm that is constantly analyzing that. So if in the first, typically it takes like a second or two to decide where this molecule comes from. So if we know that it's, if it is on a different chromosome or very, very far away from your region of interest, you can eject it right away. Now, if your molecule is a megabase long and and we can recognize it's 500,000 bases away from where you're interested in, it's kind of up to you. In the here and now nanopore sequencing, yes, you can get megabase pair molecules, but those are very, very infrequent. What we would really recommend is if you have a region of interest, expand it by maybe 10 kb. So if the molecule that you're sequencing starts within 10 kb, those are the ones that you want to keep so that it will span across the region. But if it's a megabase pair away, there's only 1 in a million chance it's actually going to reach the region of interest. So it's probably not worth keeping. It's better to reuse that activity time in order to collect more molecules over the region of interest.
All right, so I've got another question about UNCALLED. So maybe I missed it, but would UNCALLED be an extension to the classic Nanopore software for sequencing that integrates with it?
Yeah, so if you check out the UNCALLED GitHub, there's kind of detailed instructions about how you have to do it. The idea is you have to kind of deploy it in a special way into the Minnow environment. And then as you launch your run through sort of the control software for Oxford, you kind of click through the control panel and you say, yes, I want to sort of activate this mode. Admittedly, today, it requires a lot of steps. It's not fully automated. At London Calling, when was this? End of May, beginning of June, they announced that they are trying to streamline this operation. I think thanks to UNCALLED and then also some great work from Matt Luce. Oxford is really taking this sort of motor operation very seriously. So I hope that the sort of the practicalities of getting it up and running will be a lot more streamlined in the near future. But if you have questions, you get stuck, call us up. We're happy to help you out, work through those issues.
All right, so this one's from Chirag. Thank you. In applications like read mapping, whole genome alignments, etc., what fraction of overall runtime is spent on suffix array lookups?
Great question. So it all depends. It all depends. So in Mummer, there are parameters over how long do the exact seeds have to be compared to the the full dynamic programming. Depending on how long you set those seeds, seed finding could be a large percentage of the runtime, or it could be a very short percentage of the runtime. So I admit, it kind of all depends. What I really want you to take away from this is, yes, this is a way to turbocharge suffix array lookup specifically. But what I really wanted to do to speak to this audience, because we're the tool builders, we're the algorithmicists. I just wanted you to reflect for a moment that this is a very common situation where we have a strategy that in the here and now is thought of as being an algorithmic approach where we're going to follow some recursive process, some iterative process, whatnot. But maybe there's a way to frame that problem as a prediction problem. And maybe the prediction won't give you exactly the right answer, but it'll get you kind of in the right ballpark where you can avoid many, many rounds of expensive compute in order to sort of turbocharge your approach. So I'm with you to make sapling, fully a full replacement for bowtie. We would also have to have, dynamic programming and seed and extend type of analysis. And that's coming. Those are the sorts of things, of course, that we're working on now. But I really wanted to speak to this audience to kind of encourage you to think broadly about the algorithms that we're applying to see if there's some way to turbocharge them using these learned index structures.
All right. The next question is from Jorge. Can you comment on how you guys are merging and genotyping copy number variants?
Yeah, great question. So in the here and now, there's kind of two major strategies for finding copy number variants. So from long read data, especially, although there are people that try to do it from short read data, you can find the precise breakpoints, through split read alignment, where you can say, if a read is aligning along, kind of jumps back on itself, that's sort of the hallmark of a duplication. Or if it aligns and then it jumps, that's the hallmark of a deletion. So you can find deletions and duplications, with a lot of accuracy using split read alignment. And that's where Jasmine really shines, where you have these precise breakpoints. You can match it up, and you can kind of use the comparison algorithms that I just described. I will admit, though, for copy number variants, especially very large ones, if it's 100 kb region, megabase pair regions that are amplified, especially in, say, cancer, often the analysis of copy number variants is not done at that level of resolution, or not always done at that level of resolution. It's more of sort of a counting algorithm where you sort of evaluate how many reads align to a different bin in the genome. For that sort of analysis, we have a totally separate tool that we call Ginkgo. It was really developed initially for single cell analysis, although it works great also for bulk sequencing. If you have lots of samples and you have large copy number variants that you're trying to look at, I'm going to encourage you to check out Ginkgo. We have been working very hard to integrate these two worlds together, where you have sort of precise breakpoints developed primarily from long read sequencing data, and then larger copy number variants that have been recognized using coverage analysis. My senior postdoc, Sergey, has an algorithm called RCK that tries to reconcile these two sets of variants so that you can kind of resolve allele-specific karyotypes. That's what I would really recommend. Sergey has a preprint describing that. It's also described a little bit in more detail in the breast cancer organoid paper that I mentioned that will be out in Genome Research shortly. But kind of the gist of it is you need to kind of integrate and reconcile these data types. It uses something called integer linear programming to kind of satisfy a bunch of constraints about what sorts of amplifications you can have, how the variants can be connected. And it does an excellent job to reconcile these.
All right, I've got a few more questions on UNCALLED, and I think we've got time for them.
Okay.
So how efficient do you think UNCALLED would be on RNA reads, especially that RNA molecules are typically shorter than DNA, and there is a long poly A section at the beginning?
Yeah, great question. So, I mean, this is exactly the sorts of things that we're trying to investigate now. The first version, in reality, is exclusively for DNA. I guess, we haven't tried it experimentally. I think it would work okay for cDNA, but the way that the signal decoding works is really a DNA model. For RNA, you're absolutely right. There's a whole set of new challenges, right? The alphabet is different, so the signal characteristics will be totally different. There is a poly A tail, so forth. There's a few things that are sort of in our favor. The rate at which RNA translocates through the pore is a lot slower than DNA. Oxford has been working to try to optimize this. DNA passes at about 450 nucleotides per second. For RNA, it's in the neighborhood of 100 nucleotides a second. So we're going to be collecting signals. It's moving slower, which just kind of gives us more opportunity to kind of do more compute to kind of recognize where this comes from. You're right. If it's all just poly A, you're going to get basically a flat signal. There's not much we can do there. But hopefully in the first few seconds of sequencing, the first few hundred nucleotides, we can reliably identify where this molecule comes from and then do some ejection. You're right also that if all of your molecules are really short, you're not going to have great opportunities for enrichment. But nevertheless, a typical transcript, a human transcript is a few thousand nucleotides. So if we can eject in the first few hundred, I think this could give a pretty nice enrichment for those molecules we want.
And I think this is going to be the last question from Harun. Do you think that models like UNCALLED would be useful for accurately mapping k-mers, allowing some mismatches, or do you think a more complex model would be needed?
Yeah, so there's already, in the interest of time, I didn't really go through it, but UNCALLED uses an FM index at its core, and if you kind of really zoom out and look at the big picture, it works actually quite similar to Bowtie 1. If you're familiar with the history of Bowtie 1, there was kind of a really clever model where on the fly, it was allowing for a certain number of mismatches kind of in the sequence itself by kind of doing this backtracking. It's not implemented at all in the same way, but inside of UNCALLED, there is something conceptually very similar where it's going to allow for a branch search all at once. Normally when we do a lookup with an FM index, it's only for exact matching, but in an UNCALLED paradigm, it's actually considering not just sort of one path, through the FM index, it's actually exploring this whole space of possibilities. So if you just have a single nucleotide variant, small error there, it's actually quite robust to that. If you're talking about, larger structural variations, that's where things get more complicated. Although UNCALLED does a form of split read mapping on the fly to accommodate that. So I'm with you that, if your sample differs dramatically from a reference genome, there could be some issues. Although UNCALLED is actually quite a lot more robust than just exact matching, because it is doing this on-the-fly search of possible matches